So yeah, thanks very much for offering to give today's talk. Looks like a really fascinating subject, and we've got a lot of people tuning in. We'll be recording it as well, and we'll make it available to members online afterwards. So I might just hand it over to you, and yeah, really look forward to hearing about your work.
Thanks, Jamie. Sharing the screen. Oh, can you see my screen now?
You can see a white square. Yeah.
Yeah. Okay. So I'll start my presentation by basically acknowledging the territories of the Indigenous people within Canada. So I acknowledge that this particular study was conducted within the Treaty 8 land in Alberta, which is the traditional territory of the Cree and the Dene, and the unceded territory of the Métis and the ancient gathering place to many Indigenous people for thousands of years. Also, I'd like to acknowledge and respect the Lekwungen people on whose traditional territory stands within the University of Victoria, where I'm currently affiliated, and the Sanghees, Esquimalt, and the Wisanich people whose historical relationship with the land continue to this day. And just a quick advertisement of like the current project that we're working on right now. So I'm basically part of the iTrack DNA project that aims to standardize the use of environmental DNA tools, in particular qPCR-based assays and environmental monitoring. So this project aims to create targeted assays for 100 priority animal taxa and provide training and certification to different people that includes Indigenous people, consultants, researchers, and environmental managers. So one of the basic output for this project would be national standards for eDNA within Canada, and it is being led by Valerie Langlois and Karen Helping at the University of Victoria and INRS. And at the same time, I'd like to take this opportunity, to announce the fish seDNA working group that we're trying to establish within the seDNA Scientific Society, and it intends to increase the application of fish environmental DNA detection, coming from sediment samples for environmental monitoring. And together with Grayson Houston and Eric Capo, we're on the process of making a review paper for addressing difficulties in detection of fish sedimentary DNA. So to start my entire presentation, it will be focusing on this primary question wherein we want to know whether combined Indigenous knowledge and Western science in the form of environmental DNA can help us inform conservation efforts and future biodiversity assessments. In particular, for this presentation, I'll be focusing on the case of oil sands region in Alberta. So basically, this region contains high deposit of crude oil that serves as the primary livelihood for most people industries in Alberta and serves as the primary source of income for the province. And just like other industrial activities, it is expected that crude oil mining within this region can highly impact the entire ecosystem. That's why the oil sands monitoring program has been established. And this particular program produced a lot of literature about environmental monitoring within the area. And it create repositories of environmental monitoring data with 10 years worth of monitoring of on land, water, air, and wildlife. But despite this tremendous amount of available monitoring data, we're still having some difficulty in of the current state of the ecosystem, in particular aquatic ecosystem within the oil sands region, due to limited historical baseline. So with that problem of limited historical baseline data for environmental monitoring, this particular program is now tapping or finding other possible resources for baseline data. And one of this one would be Indigenous ecological knowledge from the First Nations within this region, wherein Indigenous narratives can provide past environmental conditions within this ecosystem and can serve as alternative baseline data for environmental monitoring. One perfect example would be the case of Cowper Lake and the Chippewayan Prairie First Nation, or CPFN, in this presentation. So this indigenous community holds detailed fish baseline record within Cowper Lake. So they know different fish species that has been noted from 1920s up until to present. And then in particular, this particular narrative interests us because the community knowledge holders used to notice abundant whitefish in the 1940s, and they somehow observed that it suddenly disappeared in the 1950s within this lake. And this kind of narrative can be hard to validate because the Alberta Fisheries Agency only holds fish community data earliest 1980s. So they don't have data in the 1920s or 1950s for the fish community composition for this particular aquatic body. And to address that concern, we can use some retrospective approaches with the use of, for example, lake sediment core, wherein it's basically layers of sediments that accumulate in the lake and can provide high resolution information on multiple ecosystem features. Traditionally, we're using fossilized materials. For example, for fish, we can use the fish otoliths or the fish eardrums that's scattered within or in between sediment layers. And these fish otoliths can help us identify fish species, and at the same time, it can serve as proxy for estimating particular abundance or abundance of particular fish species in the lake. But with the current advances in environmental DNA, we now have sedimentary DNA as an alternative proxy, which can be faster and less invasive to reconstruct historical fish faunal composition. So unlike the traditional approach wherein you need to use microscope and then manually scan the sediment particles and then see possible otoliths in the sediments, this particular approach or sedimentary DNA in alternative can be faster and can also provide us more accurate identification of like fish species present within the aquatic body. So in this presentation, I'll pose two questions. The first one is, can we use sedimentary DNA in reconstructing historical fish fauna using lake sediment core? And at the same time, can we infer ecosystem changes using the data that we have from this one? And then once you have the sedimentary inferred data, will it align with the historical indigenous narratives in which they have comprehensive generational accounts on the ecosystem changes within this particular aquatic bodies? So with those two questions, so my presentation is entitled Combined Use of Sedimentary DNA, or sedDNA, and Indigenous Knowledge in Reconstructing Fish Historical Records. I'm currently a postdoctoral fellow at the Helping Lab, or in Helping Lab at the University of Victoria in British Columbia, Canada. So to address those two questions, I'll follow this general methodological workflow wherein it consists of first designing robust qPCR eDNA assay, and then followed by DNA preparation and actual eDNA analysis. And I'd like to emphasize that for the sediment core, we implement randomization of the processing of the samples, wherein we used randomized processing number so that we can avoid inherent biases when it comes to possible contamination among different sediment sections. So for the sediment core collection, Cowper Lake is located in northeastern Alberta. And then we pick the deepest portion of the lake, which is in the middle part. So the maximum depth for that sampling site is around 3 meters, which I think contribute a lot when it comes to the success of detecting fish DNA coming from sediment core because of its shallow nature. So we segmented the sediment core by 1 cm interval, And these sediment sections were sent for radioisotope analysis, in particular, led to 10 for dating each sediment section. So this is followed by designing robust qPCR-based eDNA assays. So we follow the suggested workflow by Langlois 20 et al., wherein it all starts with the in silico inspection of entire mitogenome of the target species and other confounding species. Confounding species are the other fish species or other mammal species that are expected to be present in Copper Lake. So we examined their entire mitogenome and identify unique regions within our target species to ensure specificity of the assay. And then this is followed by in situ validation, wherein we're using genomic DNA extracted from both target and confounding species to test specificity of the primer and the probe. And then followed by synthetic DNA validation using G blocks so that we can create the standard curve and then calculate the limit of detection and limit of quantification for each particular assay using the EloQuant script. So for this particular study, we've used a total of 6 different assays, namely the integrity DNA assay that targets conserved region within the plant chloroplast so that we can test viability of the extracted DNA from the sediment. We implement this one because we're worried that some of the DNA that will be extracted can be degraded because of their old nature. So once we use plant DNA because they are quite ubiquitous when it comes to environmental samples, so making them a good marker for internal control for viability testing. We also tried to design, in the lab, we designed a general fish assay, which is targeting a conserved region within the mitochondrial genome of the fish. And this will be used for screening sediment sections, whether they contain general fish DNA or there's no fish DNA within each sample. And then we'll proceed with species-targeted assays. The first one would be the lake whitefish, or the e-cockle one. And then we also have another assay for another species of whitefish, which is Cisco. I highlighted this too because these are the species of interest for the indigenous community. So remember that the indigenous community used to observe abundant lake whitefish in the lake. So we're thinking it can either be lake whitefish or Cisco. So that's why we have a species targeted assay for those two. And then we also have an assay for northern pike, which will serve as positive field control because this particular species has been noted to be present in the lake for the longest period of time. And then we also have a specific assay for walleye, which will serve as negative field control because this species is not noted or not present in the lake. So just to give you a quick overview on the sensitivity of the assays designed in this study, I'm presenting the limit of detection, which is the lowest possible DNA copy that can be detected by each assay. So it simply shows that the assay, it's either highly comparable with other fish assay in literature, or if not comparable, it's even like much more sensitive in terms of limit of detection. And same thing when it comes to limit of quantification. So in comparison to other fish eDNA assays in literature. So it's either comparable or much more sensitive in terms of LOD or LOQ. And this is followed by careful DNA preparation because one of the difficulty in detecting fish sedimentary DNA is that it's either your DNA are highly degraded or there are a lot of PCR inhibitors. So we start by using 2 grams of sediment samples and then extracted the DNA using the Norgan soil extraction kit that has its own built-in column for the removal of PCR inhibitors. After extracting the DNA and eluting it in 2 ml, we tried to reconcentrate the DNA by precipitating it using sodium acetate and ethanol approach. And then this concentrated DNA is then we did another set of PCR inhibitor removal using a column-based approach so that we can ensure that there's a total, or like we can remove as much PCR inhibitors as possible when it comes to the DNA samples that we'll be using. And then it's followed by DNA viability testing using integrity DNA assay. So showing you the result for the viability testing, as you can notice, we've dated the sediment core sections from 1905 up until to 2019. And then on the y-axis, I'll be showing you the quantified DNA copies for the plant chloroplast, or the target region within the plant chloroplast. So we've detected the target region from all sediment sections. And I'd like to emphasize that the highest DNA copies were noted in the youngest sediment core sections. And then in terms of detection frequency, we've used a total of four technical replicates for each reaction. And as you can note on the figure, like we had like amplification out of like all technical replicates for each reaction coming from all the sediment core sections. And then another layer is that all of the DNA copies calculated in this particular analysis are above the limit of detection and limit of quantification for this particular assay, thus increasing our confidence that the DNA that we have for the succeeding eDNA analysis are viable and not degraded. So to proceed, we'll go with the actual eDNA analysis using the general fish assay and the species targeted assays. So I'll present the result first for the general fish assay. Same figure. We have the sediment core sections dated 1905 to 2019. And then we have the quantified DNA copies. We've detected fish DNA from the oldest sediment core section up until to the youngest sediment core section. But same as with the plant chloroplast DNA, we've detected the highest concentration of fish DNA on the youngest portion of the sediment core. This can be attributed to the possible integrity of younger DNA in the sediment core sections as compared to the older section of the sediment core, wherein there's possibility of degradation and highly fragmented DNA in those samples. And then also, I'd like to emphasize that the DNA copies for this particular assay are all above the limit of detection and limit of quantification for this particular assay. So on the later part of the presentation, I'll emphasize why I keep on telling that all of the DNA copies calculated for each assay are above the limit of detection and limit of quantification. So for the fish targeted assays, so we only detected positive amplification from northern pike, which is the positive control for this lake. And then we also detected Cisco, which is one of the lake whitefish species that we are really interested. And we don't see any amplification from lake whitefish and walleye, which is the negative control. For this assays, I'd like to pinpoint that we calculated lower DNA copies as compared to like the general fish DNA and the plant chloroplast DNA. So this gives us an idea on like possible abundance, or it's just basically it's not to say if like this is correlated with the fish abundance, but maybe like can be attributed to the high spatial heterogeneity on the distribution of fish DNA within the sediment layers. So basically for this one, since we're observing lower DNA copies, to make sure that we can avoid false positive detection for the fish, this is where your limit of detection and limit of quantification play a very important role. So if the DNA copy that we calculated for a particular sediment core sample is lower than the limit of the detection, we are considering that one as negative result. If the concentration of DNA that we calculated is above the limit of detection and better above limit of quantification for that particular assay, this gives us much more confidence that this particular amplification is more probably like more accurate representation for the presence of this particular species in the sediment core section. So for this one, it gives us an idea that northern pike is really present at different time frame within the sediment core section, while for Cisco, it has been only noted from 1928 up until to 1943 for the sediment core sections. And when it comes to the consolidation of the indigenous knowledge, we have collaborators who went to the territory of like the Chippewayan Prairie First Nations and then interviewed some community members. In particular, we focused on the elders that serves as the knowledge holders for this entire community. And to summarize everything in consolidating sedimentary DNA data and the indigenous knowledge coming from the community that we are partnered with. So the sediment sections, as I've mentioned, were dated from 1905 to 2019. The bar shows the standard error for those date estimates. And then we also tried to estimate sedimentation rate for each particular section using cesium and RA radioisotope analysis. And then, as you can take note, there's a rapid increase in sedimentation rate within the 1940s. Sedimentation rate can give us an overview on the possible human activities within the lake because sediment suspended in the water column may be attributed to possible human activity. For example, runoff coming from the surroundings of the lakes or terrestrial inputs when it comes to possible agricultural or logging activities or mining activities within those time period. And we inserted the result for the general fish DNA, wherein it's totally distributed from the oldest to the youngest sediment core sections. We detected amplification of northern pike, which is the positive control for this lake, with less frequency or with less occurrence within the sediment core sections, but it's detected from the oldest up until to the most recent sediment core sections. We also detected one of the lake whitefish species, which is the Cisco, or whitefish species, which is the Cisco, and it's detected from 1920 to 1940s, which is somehow aligned with the narrative of the indigenous communities that they've noted this one in the earliest 1950s, and it started to decline in terms of population in 1950s. And then we also align this particular data with historical land use. The first one would be the active human migration within this region because this is the time period wherein they are trying to increase the crude oil mining within the oil sands region close to the lake. And then the community also have a narrative that there used to be a natural landslide, or they noted natural landslide on the eastern part of the lake, which somehow aligned with the increase in the sediment rate or the spike in sedimentation rate within that particular time period, and possibly can also be the reason for the decline in the population of the Cisco. And then the recent increase in sedimentation rate is aligned with the other human-mediated activities around the lake, which is industrial development within the region, forestry and logging, and then oil sands activities, of course. So basically, this summarized all the data that we inferred from sedimentary DNA and the radioisotope analysis and the community knowledge that we collected from our partner Indigenous community. So going back to my first question, can we combine Indigenous knowledge and Western science in the form of environmental DNA in informing ecosystem health monitoring program? So basically, sedimentary DNA data can provide us empirical data and much more accurate measurement of the trends when it comes to the fish community. So for example, lake whitefish or Cisco were detected from 1920 to 1948. Strong amplification signals may imply that there could be like high abundance of this fish in that particular region. So, but like as a disclaimer, eDNA copies, it's still hard to correlate it with the fish abundance. On the other hand, we have the indigenous knowledge, specifically the Chippewayan Prairie First Nation narrative, that they observed whitefish species in the early times, and then it started to disappear in the 1950s. There's a natural landslide in the eastern part of the lake in the 1950s that might be the reason for the disappearance of this species in this particular aquatic body. So in general, indigenous knowledge can provide us generational observation of ecosystem changes that we might have missed because we don't have earlier ecosystem monitoring data for those time periods. And once you combine this two, it basically provides a more comprehensive understanding of the long-term human-mediated ecosystem changes in the water bodies that you're studying, in particular for this one, the Cowper Lake in the oil sands region in Alberta. So to conclude, I think it's somehow safe to assume that the combined use of sedimentary DNA, in particular fish sedimentary DNA, and indigenous ecological knowledge can be an effective approach in assessing current and historical historical ecosystem health, in particular for this one, fish communities. And what's next for this project? We're currently in the process of analyzing other lakes within the oil sands region. So we have collected sediment core samples from 5 more lakes in this area. And then we're also analyzing heavy metal profile from the sediment section so that we can have more environmental parameters to infer and correlate with the presence absence of the fish species for each lake. So with that, I'd like to acknowledge all the help coming from this team, in particular the funding from the Oil Sands Monitoring Program. So this is a combined effort from University of Victoria, the Canada Environmental and Climate Change, and then Alberta Environment and Parks, and of course our Indigenous partners from the Chippewayan Prairie First Nations and the Cold Lake First Nations in Alberta. And I think that's all. So thank you, guys.
Thanks very much, Louie. That's really fascinating talk. Does anyone have some questions for Louie?
I have a question. Hi, Louie. Thank you for your presentation. That was really good. I would just want to, I don't know if you mentioned that, maybe I missed it. The length of the targets, of your targets, how much was it?
For this one, for when we are designing for qPCR based, we are eyeing amplicon target from 70 to up until I think 350. So basically for all these assays, I'm not particular like the exact base pair amplicon size, but it's basically within the 70 base pairs up until the 350 base pairs.
Okay. And did you test this assay with water samples from the same lake?
Yeah, so for our workflow in the lab in terms of validation or validating qPCR assays, we start with in situ validation, wherein we are using actual genomic DNA extracted from the tissues. And then another part of our validation is that we are conducting field validation. So we didn't test this assay for detection of this species using lake water from Cowper, but we basically did field validation from using water samples from other aquatic bodies that we know that this particular species are present.
Okay. And the last question, sorry.
No
To design this assay, did you use published mitogenomes?
Oh yeah. So for this one, we basically used available mitogenomes for coming from the GenBank. So just downloaded it from NCBI.
Okay. Did you had any issues with prokaryotic contaminations in those genomes?
Do you mean like bacterial contamination?
Yeah. Yeah.
I'm not particularly sure because those sequences, like we just downloaded them from NCBI. But like uh, for our pipeline, it just depends. Like for the mitochondrial sequences, if it's available in the GenBank, we'll just download it. Considering that the sample used for sequencing, it's collected within North American region or North
Mm-hmm.
America region because the problem sometimes is that some mitochondrial genome were like sequenced using samples from Asia, and that's when we're having problem when it comes to specificity due to like
Yeah.
population differentiation of the species. Yeah.
Yeah. Okay. Thank you.
Yes,
Brian?
Yeah, curious on the depth of the other lakes that you're sampling in northern Alberta.
Mm-hmm.
Cold Lake, that's a lot deeper, right? Than some of the other ones.
Yeah. Mm-hmm.
So do you have a hypothesis saying that basically the cold water fish, such as what whitefish or Cisco, are going to be in those lakes and have survived? Is it a test
Mm-hmm.
of a climate hypothesis or is it a test of something else?
Yeah, that's all that's really possible, especially the input like the rivers across like this within this region are also connected in terms of like the tributaries. So that's possible. And yeah, so but basically for this one, we're not in like in general for this project, our objective first is like to test the feasibility of the method because in general for fish sedimentary DNA, to be honest, it's like we're still having a lot of difficulties for
Right.
troubleshooting the actual method because it's really hard to detect fish DNA from the sediment samples. Yeah, so far we're still focusing on the methods, but
Okay.
we're definitely will explore like the ecological aspect of this project when it comes to fish communities and fish population within this region.
Yeah, because you mentioned that the lake that you sampled was 3 meters deep, right?
Yeah.
Which is sort
Uh,
of unusual habitat for Cisco, isn't it?
mhm.
Yeah.
Yeah.
Whereas
So
Cold Lake is like 100 meters deep.
yeah. So
Yeah.
I somehow mentioned that one because I think that's one of the reasons why it has nothing to do with the biology of the fish for now, but it's one of the reasons maybe why we're having success of fish detection from the sediment core because it's very shallow.
Yeah.
So the transport of the DNA from the water column to the sediments, it should be faster. And considering the higher sedimentation rate for those time period, so that right now what I'm thinking is that basically contributed a lot with the integrity of the DNA preserved within the sediments. And my
Okay.
hypothesis, like for example, for Cold Lake, which is 120-meter deep. So my hypothesis that we'll be having difficulty detecting fish DNA for that one because it will take time before the DNA from the water column to be transported to the sediments.
Okay.
Yeah, I'm
Thank you.
sorry. I'm not sure if I answered your question.
I think you did. I was more wondering if one of the reasons why you might have lost the fish or the whitefish in your lake
Mhm.
is if you have a 3 meter lake and it's warming up over time and temperature
Yeah.
changes, you have a big impact on a cool water fish if you're whitefish,
Mhm.
or if you're
Yeah.
Cisco, you're a cold water fish. So
Yeah. Mhm. Yeah, that's really possible. Or another thing that we're thinking is that since there is a rapid increase in sedimentation rate, there could also be like possible or like gradual change in the depth of the lake through time. So like from a deeper lake to like more shallow lake due to the like terrestrial runoff from the activities around the lake. yeah, that can also be a possible reason.
Okay. Nope. Sounds good. Thanks.
Thank you.
I have a question kind of also related to the ecology of the fish.
Okay.
In the diagram you showed, the period where you had Cisco turning up, there was no northern pike and vice versa. They sort of seemed exclusive of each other. Is that to be expected from their ecology?
I'm sorry, can you repeat your question?
Do Cisco and northern pike live at the same places? Do they have the same ecology? And if they don't, might that explain why one is present and the other isn't? And there's kind of they don't occur in the same sediment layers together.
Based on my limited knowledge on North American limnology, because before going to Canada, I used to study tropical lakes in the Philippines and Taiwan. So I'm very new with the temperate lakes. But based on some literature that I've read, there are some few lakes wherein we can see both northern pike and Cisco coexisting in same lake. So yeah, my I think in relation to your question, my only concern when it comes to this data set is that we're really interested to see much more older sediment core section, because we want to know whether this Cisco, if it's just introduced or it's native within this lake, because we don't know, we don't have a data if it's present before 1905. So the oldest sediment core section that we have is 1905. So we don't know that if this observation, it's mainly due to observation due to the introduction of Cisco within this time period, because as you can note, within the historical land use, this is also the time we're in. There's active human migration within this region because they're trying to increase the crude oil mining within the area. So it might be also correlated with human migration, and they all of a sudden they introduce Cisco in the lake. That's why it also didn't lasted for a while, because it's not really an optimum Copper Lake is not an optimal habitat for this type of species. Yeah.
Okay. Do we have any other questions?
I just have another really quick question. When you dated the sediment core, is that alpha dating or gamma dating? And do you have cesium on this lake as well?
Yeah, we have cesium and then RA for the analysis.
Does the cesium show a nice peak in this lake, or is it too organic?
Yeah, we're seeing a really nice peak for cesium for this one.
Okay. And it shows up right around where you're dating 1963 or so?
Yeah. So for this one, we're still digging much more data, much more analysis when it comes to the radioisotope analysis. So that's why I wasn't able to present the data. But I'm thinking to make one separate figure for the radioisotope analysis for the publication of this data.
Excellent. Thank you.
Yeah. So thank you for this opportunity.
Yeah.
To present our work.
Great. Thanks so much. And if there's no further questions, we might end it there. Yeah. Thanks so much for your talk, and it will be made available online, and we will send out a link to it in the next few days.
Okay. Thanks, Jamie. Thanks, everyone.